you very much, Françoise, for this kind introduction. I'd like to start by thanking the IS for the invitation. It's a great pleasure to be here and a true honor to address this audience. So since the isolation of HIV in 1983, we have better weapons to fight HIV infection, and we have better drugs, more drugs. And if you compare the efficacy of these drugs in the pre-antiretroviral therapy era to now, clearly they show higher potency and reduced toxicity. And more importantly, the number of pills the people living with HIV have to take per day has been considerably reduced. And this is clearly a great scientific success. And this great scientific success has also translated into clinical benefits for the people who are living with HIV and AIDS. And if you look at the expected survival of a 20-year-old person living with HIV in a high-income country between 1995 and now, you would see this dramatic increase in the number of years people can expect to live with HIV drugs. And this life expectancy is actually quite close to the one you would see in the uninfected populations. So clearly, this is a great success. And one question you might ask is, why do we need a cure? Why do we need to do better than antiretroviral therapy that works very well? Well, I think there are different answers to that question. So first, antiretroviral therapy do not eradicate the virus. So it greatly reduces the amount of virus in the body, but the virus is still there. And because the virus is still there, other issues either appear or persist. And I'd like to take a few minutes to go through them. The first one is that the criminalization and the stigmatization of people living with HIV still remains in 2015 in most of the countries in the world. People living with HIV face many issues: criminalization, stigmatization, in their family and at work. For that first reason, a cure for HIV is something that we all want to develop. A second reason why we need a cure for HIV is that access to antiretroviral therapy for everyone is a great challenge. There are many people in need of antiretroviral therapy in the world. Around 40% of the people who need drugs don't have access to it. So again, access to ART for all is a formidable challenge. The third reason why we need to do better than ART is that those treatments are actually quite toxic. And as you know, these drugs have to be administered indefinitely for your lifetime. And we know that they may induce some side effects. And more importantly, we don't know what are the long-term effects of these drugs for more than 20 years. So if you need a fourth reason for why we need a cure, I'm going to give you one more. And this is money. And here I'm taking the example of Canada. And on this slide is summarized what those new infections are going to cost to the healthcare system. HIV still generates enormous cost for Canada in terms of human suffering, job loss, and its related economic impact and financial burden on our healthcare system. And you'll see it's not only the price of the drugs, it's also labor productivity and the quality of life of people living with HIV and AIDS. So this is another good reason why we need to do better than ART and develop a cure for everyone. Because of course, if we had a cure, we would eliminate the virus, and these issues would instantly disappear. So as I just mentioned, antiretroviral therapy does not eradicate HIV. And this is what we see in many, many people who have been interrupting treatment during the past 20 years. The virus comes back quite rapidly after treatment interruption, again, in the vast majority of the people who stop antiretroviral therapy. So where does HIV hide during antiretroviral therapy? Because again, those drugs reduce the level of virus to undetectable levels by viral load, but clearly the virus is still there. So it's quite challenging to try to identify the places in which the virus persists. And we know from the work of many people in the field that several parts of the bodies can harbor latent HIV or some low levels of replicating virus, in particular in the brain, in the lymph node, in the gut, as well as in the genital tract. So this is at the organ level, but we can also have a look at the cellular level, because those organs obviously are made of cells. And the best characterized reservoir are CD4 T cells. So you know CD4 T cells are the main target for the virus. but they are also the main cells in which the virus can persist during antiretroviral therapy. And the way the virus persists during antiretroviral therapy, there are actually different ways. So the virus can still replicate at low levels, and this is what we call the active viral reservoir. And this is probably true in particular in tissues, because we know that some of the drugs we're using do not completely diffuse in either the brain or in the lymph node, so that would allow the virus to still replicate at low levels in these organs. In addition to this active reservoir, there is the existence of this latent reservoir that was actually discovered by 3 independent teams back in 1997. And this latent reservoir is made of cells in which the virus is silent. And this silent virus, or these cells that harbor silent virus, can persist for a very long period of time. And it has been estimated that the half-life of the reservoir is 44 months, which means that more than 70 years of antiretroviral therapy would be needed to eradicate this latent reservoir. So extremely long-lived, and actually the cells that harbor the latent reservoir are also extremely long-lived. And they can also undergo low levels of cell proliferation. And this is another mechanism for the virus to persist during antiretroviral therapy. This T-cell proliferation has been reported last year by 4 different teams who very elegantly showed that proliferation of latently infected cells is a major mechanism for HIV to persist. So, as I just showed you, there are many, well, I would say 3 different or main mechanisms by which the virus persists during antiretroviral therapy. And this comes from the fact that all CD4 T cells are not the same. And on this slide, I'm showing you what we call the memory phenotype of the different memory CD4 T cells. So from the naive cells that are in green here to the family of memory CD4 T cells. And from the left to the right, you're going to from the less differentiated to the more differentiated. Stem cell memory cells, central memory cells, etc. And those cells have different capacities to produce either cytokines or to be activated or to self-renew. So it is logical to think that they will actually contribute to HIV persistence through different mechanisms. So back in 2009 with Rafik Sekali, we started this study in which we asked the question, where are the cellular reservoirs for HIV and what are they? So we actually took lots of biological samples from people on triple therapy, and we sorted those different subsets to identify the subset in which the virus was hiding. And what we found is that the virus is hiding mainly in the memory cells, either the central memory cells, TCM in blue, transitional memory cells, and effector memory cells. More recently, the group of Matthias Lichterfeld and Maria Bezen identified another cellular reservoir, the stem cell memory. And I'd like to take a minute to talk about those. So in their paper, Matthias and Maria reported that though this small compartment of CD4 T cell is also extremely long-lived. And if you look at the contribution of this subset over time, it actually increases, suggesting that this is probably the most persistent reservoir for the virus during antiretroviral therapy. And this has also been shown by the group of Yassine Taoufik in France, who actually estimated the half-life of this cellular reservoir and found it to be extremely persistent as well. So that might be the one we want to really try to eliminate. So on this slide, I'm showing the 3 main barriers to an HIV cure. So first, HIV persists in tissues. Second, latently infected cells are rare and undistinguishable from uninfected cells. And latently infected cells are diverse, like I just told you. So what do we need to do? We need eradication strategies that can reach the tissues. We need eradication strategies that should be specific, and we need eradication strategy that should target all latently infected cells. There are 2 main types of cure that have been proposed. The first one is the sterilizing cure. This is the ideal model in which you would eradicate all trace of virus from the body. The second type of cure, which is probably more realistic, is what we call a functional cure. And in that case, you would probably try to also reduce the size of the reservoir significantly and then boost the immune system so it can control some events of reactivation from the latent reservoir. And actually, we have different examples of cure that have been reported in the literature, or functional cure or even sterilizing cure. So again, this is what happened in the majority of people on triple therapy. When they stop ART, the virus rebounds usually in less than 2 months. We have the extreme case of Timothy Brown, also known as the Berlin patient. And as you know, Timothy Brown is free of HIV for more than 7 years now, after being transplanted with protected cells from HIV infection that are CCR5 deficient. And between those two extreme cases, we have what we call the Boston patients that have been reported by Tim Henrich and Dan Kuritskas. And those two individuals have also received bone marrow transplant, but those cells were not CCR5 deficient, so not protected to HIV infection. And unfortunately, both individuals rebounded 3 and 8 months after cessation of antiretroviral therapy. You all know the case of the Mississippi child reported by Debbie Perso and Catherine Lazuriaga. This little girl was treated just after birth and was able to control naturally HIV infection for 28 months, but unfortunately eventually rebounded. And finally, we have these, I think, fascinating cases of the Visconti and Spartak cohorts of post-treatment controllers, people treated during acute infection who were able to control infection after treatment interruption. So if you look at that, there is a common point between the Mississippi child and the Visconti and Spartak studies, and all these individuals have been treated very early in infection. And in collaboration with the military HIV research program and the Thai Red Cross, and in particular Jintanat and Environich, we actually looked at the impact of early ART intervention on the size of the reservoir. And in black, you can see the size of the reservoir after treatment initiation in people who are chronically infected with HIV. In red and in green are people who have been very recently infected. And as you can see at baseline, the reservoir is already smaller than the one you would see in the chronically infected patients. But more importantly, their reservoir decreases very rapidly after treatment initiation when ART is initiated very early on, indicating that very early ART dramatically reduces the frequency of cells carrying integrated genomes. And we also looked at the subset to be more sensitive. I told you before that central memory, transitional memory, and effector memory cells are the major reservoir for the virus. And in these individuals who have started ART very early on, after 2 years of ART, we actually did not find any trace of virus in this subset. That does not mean that the virus is still there, but it's at extremely low levels. So this early ART also has a clinical benefit. And here I want to take a minute to speak about the Visconti study from Azir Saysirian and collaborators in Paris. So they identified 14 post-treatment controllers originally. And this individual started ART within 10 weeks after primary infection for a median time of 3 years. And after cessation of ART, they've been able to naturally control the virus for an average time of 9 years. And we still do not exactly understand how that works. But here's a typical patient on this graph. And more importantly, when Azier took blood from these individuals, he was able to recover replication-competent HIV. So the virus is still there. It's controlled by the immune system. And I think we have a lot to learn from these extraordinary cases. So how do we cure HIV? That's the big question. So the first thing you can do is to limit the establishment of the reservoir by giving ART very early on, and I just show you some evidence of that. Now, for the majority of people who have not started ART during acute infection, what do? We have different strategies to try to reduce the size of the reservoir. The first one is to render uninfected cells resistant to HIV, to deplete infected cells, or to flush out the latent reservoir. So let me start by the first strategy. And this is from the Pablo Tebas and the Sangamo study that was published last year. So in that study, they collected large volume of blood from individuals on suppressive therapy. They enriched for their CD4 T cells, and they transformed their CD4 T cells in vitro with a zinc finger to make them resistant to HIV infection. And so those resistant cells are then reinfused in the participant, and they then monitored the size of the reservoir over time. And it was quite impressive to see that 6 of the 9 participants showed a significant decay in the size of the reservoir after this procedure. And very recently, Jumana Zaidan and Rafik Sekali actually looked at the possible mechanism for this observation, and what they found is that those CSCM cells that I was talking about that are supposed to be a very long-lived reservoir for the virus, they actually expand after infusion of the product, but importantly, they are largely protected from HIV infection because they have been mutated for CCR5. So those cells probably expand in vivo and dilute the latent reservoir, and maybe there is also an active mechanism that contributes to the decay of the reservoir. Another strategy to eradicate the virus is to deplete reservoir cells. And this strategy has been proposed by the group of Andrea Savarino in Rome. And he identified this drug, which is a gold-derived compound called oranophine, that can be administered. And this drug actually induces differentiation of those latently infected cells and push them towards apoptosis. So I won't go into the details, but Andrea generated some proof of concept data in the monkey model, in which he gave oranophine to these animals, and he was able to induce not a sustained remission, but you see, those animals are able to control from time to time their viral load. And interestingly enough, when Andrea depleted CD8 T cells from these animals, the virus rebounded, indicating again that the immune system is extremely important to control this viral reservoir. So the last strategy to try to eliminate the virus is what we call the shock and kill strategy. And in that strategy, you'd like to use a latency reversing agent, so something basically that will wake up the virus. And if you do so, this cell will start to produce viral particles, but because antiretroviral therapy is maintained, those viruses won't be able to reinfect new cells and to disseminate. So what you are hoping for is that this virus that is produced by the cell will either directly kill the cell, or because this cell will be visible to the immune system, it will eventually be eliminated. So basically, what you want to do is to reactivate HIV production in those cells in which the virus is sleeping. And this is quite complicated because the molecular mechanisms that are responsible for latency are numerous and complicated to revert. And on this slide are listed some of the possible drug targets that are now being explored to achieve this. So they have been tested in vitro in cells from patients, and this is a paper from Greg Laird in the group of Bob Silicano, and they tested single latency reversing agents that are targeting those different targets that I just talked about. And interestingly enough, it's only when you combine them that you start to see an effect. And the reason for that is because those mechanisms of latency, again, are numerous, and targeting only one of them is not sufficient. So pretty much like we do with antiretroviral therapy, it is very likely that a curative strategy will need to combine several molecules that target several mechanisms of HIV latency. And this strategy has actually been tested in vivo, first by the group of Dave Margolis and Nancy Archin in 2012, and they administered this drug, which is called Vorinostat, that has shown some effect at reversing latency in vitro. And when they did so in suppressed individuals on triple therapy, they actually find an increase in the level of transcription, which is what you want to see. So it seemed to work. And then different groups have actually used either the same molecule in multiple doses, the group of Sharon Lewin, or other molecules from the same family, like panebinostat and romidepsin by Thomas Rasmussen and Ole Sogard. And they all found that this molecule can increase the level of viral transcription in this latently infected cell. So it seems to work pretty well, and you can induce HIV transcription. The bad news is that that does not impact the size of the reservoir. And in none of these studies, the size of the reservoir was reduced significantly. So the reservoir is still very stable. Even though you were able to induce some viral transcription event, there is no decrease in the size of the reservoir with only one single molecule. So this is quite depressing, but I think not only, we're actually learning a lot from these experiments. And what we learned is that trying to just induce viral production is not enough. And what you need to do is to also facilitate the clearance of infected cells. And you also probably need to enhance immune control. And this is why the cure strategy needs both virologists and immunologists. So let me give you an example of something that might do both, reactivate the virus and boost the immune system. And this comes from the study performed by Gilead and Jim Whitney. And this TLR7 agonist, which is a molecule they recently developed, can be given to non-human primates that are virally suppressed on ART. And as you can see on this slide, after the 4th and 5th administration of the drugs, they started to see some viral bleeps, which again is what you want to see in a reactivation strategy. And all the animals actually showed these bleeps after the 6th administration of the drug. And what is, I think, fascinating is that at the same time, CD8 T cells got activated. So again, this drug might be able to impact not only the viral reservoir, but also the quality of the immune system, which is something that you want to do. Finally, I'd like to talk about these immune checkpoint blockers, which is something that we have worked a lot in my lab and in collaboration with many others in the DARE collaboratory. Those immune checkpoint blockers negatively regulate T cell responses. They actually contribute to immune exhaustion, not only in infectious diseases, but also in cancer. And I'm listing here some examples, PD-1, LAG3, TIGIT, and CTLA4. And what Rémi Fromentin in the lab found is that cells that expressed these molecules are actually enriched for the latent reservoir. So if you want to selectively target the latent reservoir, you might want to target the cells that are expressing those receptors. And these are not only biomarkers, they actually have a role in the maintenance of the latent reservoir. And this is an experiment that Rémi did. Here are cells from an individual that is treated on suppressive therapy. There is no viral production because those cells are latently infected. You can induce viral production with a positive signal. But interestingly enough, when you engage PD-1, one of these molecules, at the surface of the cell, you will abrogate reactivation. So immune checkpoints are expressed at the surface of infected cells and inhibit viral reactivation from latency. So what you want to do is exactly the opposite, to block these immune checkpoint blockers to reactivate HIV from latency. And this is something that we have tried to do in vitro, and I'll show you the data in a minute. So this is what we did, and again, this is an experiment performed by Rémi, and when he had this anti-PD-1 antibody, which is available and actually given to cancer patients right now, he was able to see an increase in the level of viral production in the test tube. And Sharon Lewin just published an evidence that this strategy might also work in vivo. In one cancer patient, she was able to find that the anti-CTLA4 antibody, which is another immune checkpoint blocker, had an impact on viral reactivation. So I'd like to finish with this. How do we go from care to cure? Well, the way is long and arduous, and we have many steps to climb. The first one is HIV replication. The second one is HIV that persists in tissues. And the last one, which is the biggest one, so 2 steps, is the latent reservoir. So we have weapons to try to climb all of these steps. The first one, HIV replication, of course, we have antiretroviral therapy that works pretty well. And if you're taking antiretroviral drugs, your viral load would be controlled in the vast majority of the cases. So what do we do with HIV in tissues? Well, we know that there are some drugs that have a better ability to penetrate in tissues. So you might want to actually optimize the regimens to be able to target those persistent reservoirs in which the virus still replicates. What do we do with the latent reservoir? First, ART in acute infection is key. This is, to my knowledge, the only and most efficient way to significantly reduce the size of the latent reservoir, and this has been shown by many different independent groups. So ART in acute infection is really key. Then what do we do with the latent reservoir that is established anyway? I think we need to combine different strategies that would reactivate the latent reservoir, but this will probably not work if we don't boost the immune system at the same time. And I think the TLR7 strategy or the immune checkpoint strategies are one of the most promising ones that we should probably test in people living with HIV pretty soon. So finally, I'd like to thank all the people who have contributed to this presentation, my colleagues at the Centre de Recherche du CHUM and Université de Montréal, mainly Rémi Fromentin and my other colleagues. At Case Western University, Rafik Sekali, our wonderful collaboration with MHRP and the Thai Red Cross, and in particular, Jintanat and Anvoranich. I'd like to thank Steve Dix and Sharon Lewin for their help in putting together this presentation. I also would like to thank the funders and the Towards an HIV Cure initiative from the IIS. And finally, I'd like to thank the study participants who have generously contributed to this study. Thank you very much.
Please welcome Dr. Gregory Taylor. Dr. Taylor is the Chief Public Health Officer of Canada.
Merci beaucoup, Dr. Chauman. Very thoughtful presentation, very compelling evidence. I'd like to echo Dr. Barré-Sinisi's comments earlier. It's certainly a privilege to co-chair this morning's session. Our next speaker is Thanya Wee Putakit. Dr. Putakit is an associate professor and pediatric infectious disease specialist from Thailand. She has been conducting pediatric HIV research for more than 10 years. She is a leader in pediatric HIV treatment and care in Asia and leads several multi-center research projects in Thailand, Cambodia, and Indonesia. This morning she will discuss HIV comorbidities in children and adolescents. This is an especially interesting presentation for Canada, as maternal, newborn, and child health are one of our priorities for international development. It's my pleasure to welcome Thanya Wee to the podium. C'est avec plaisir que j'invite Thanya Wee sur la tribune.